All right, welcome back everyone for more biotechnology. So we've spent plenty of time talking about solutions and solution making and some of the calculations that are involved in that. So next we need to talk about something that is important considering the fact that almost every time you make a solution, your solvent of choice is going to be water, and that is going to lead us into a discussion on the pH scale. So water, which we've already established, is going to be your solvent almost every time you make a solution, is going to infrequently ionize into hydrogen ions and hydroxide ions. So basically, if you kind of put those two things together, you can kind of see how you end up back with a water molecule. So water, like I said, does not do this very often at all. Maybe 1 in every 10 million water molecules will actually do this. But they will do this, and there will be a certain concentration of hydrogen ions and hydroxide ions in any aqueous solution. So what pH is a measure of hydrogen ion concentration that falls on a logarithmic scale, a log base 10 scale. So the equation for calculating the pH of a solution is thus: pH equals negative log base 10 of the hydrogen ion concentration, where those brackets around the hydrogen ion is meant to be the molar concentration of hydrogen ion in your solution. So pure water, meaning it has no added solutes whatsoever, will have a pH of 7, which is considered neutral on the pH scale. As the pH decreases, the hydrogen ion concentration is going to increase. So this is going to seem a little backwards before you really kind of start getting comfortable with this. As the pH decreases, the hydrogen ion concentration goes up. So anything that has a pH of less than 7 is what we consider to be acidic, meaning it has a greater and greater and greater concentration of hydrogen ions as the pH gets lower and lower. Similarly, anything with a pH of greater than 7 is what we would consider basic or alkaline. So if you have a basic solution, then your hydrogen ion concentration is relatively low and your hydroxide ion concentration is relatively high. So that's kind of the balancing act here. At the neutral pH of 7, the hydrogen and hydroxide ion concentrations are equal to each other. As the pH becomes more acidic, you shift the balance in favor of the hydrogen ions, and as the pH becomes basic, you shift the focus to the hydroxide ions. So in this table here, you can get kind of a rough idea of what the relationship is between the pH and both the hydrogen and hydroxide ion concentrations. So here you can see the concentrations, the molar concentrations of both ions with pHs of 0, 1, 2, 3, and all the way up to the most basic on this scale of 14. So the thing to make sure that you understand here is that we are dealing with a logarithmic scale. And because this is a logarithmic scale, a difference of 1 pH unit actually signifies a rather large difference in hydrogen ion concentration, a tenfold difference as it were. So if you compare, say, the hydrogen ion concentration at a pH of 7 with the hydrogen ion concentration at a pH of 6, a pH of 6 is 10 times more acidic than a pH of 7 because it has 10 times as many hydrogen ions in solution. A 2-unit difference would signify a 100-fold difference, so a pH of 5 would be 100 times more acidic than a pH of 7. A pH of 14 would be 1,000 times more basic than a pH of 11 because there is a 3 pH unit difference there. So just because there is a fairly small difference in pH values, that does not necessarily mean there is a very small difference in actual hydrogen ion concentration. You have to remember this is a logarithmic scale, so differences are going to be bigger than what they seem. So here you can actually see a figure that is actually not from your book. It's actually from a physiology book that I teach out of. But this just kind of gives you a general idea of the pH of some household products that are basically aqueous solutions. So you're going to see things like milk and coffee and grapefruit juice and lemon juice and various sodas, seawater, cleaning solutions. So you can get kind of a basic idea, no pun intended, of what the pH is of some common things that you're likely to encounter throughout your lifetime experience. So as an example for how this logarithmic scale works, if you look at your blood, which has a pH of 7.4, which is just ever so slightly basic, and you compare it to the pH of urine, which is about 6.4, because there is a 1 unit difference between them, we would say blood is 10 times more basic than urine, meaning it has, you can either say 10 times as many hydroxide ions or 10 times fewer hydrogen ions. And the comparison works the other way. You could also say that urine is 10 times more acidic than blood. Another example, if you compare coffee and pure water, we're comparing the pH value of 5 for coffee to 7 for water. That's a 2 unit difference, so we would say that coffee is 100 times more acidic than water here. So when you're calculating the pH value, you only have to use that equation that was on the previous slide. So you're going to need a scientific calculator that is capable of logarithmic functions in order to do this. So if we ask ourselves, what is the pH of a solution that has a hydrogen ion concentration of 0.0005 moles per liter, well, all we would have to do is plug this in. Negative log base 10 of 0.0005 would give us a pH value of 3.3, so that would be fairly acidic. And since we have had prior conversations about different methods of representing concentration, such as molarity, mass per volume, percentages, and things like that, make sure you understand that only molar concentration works for this particular formula. So it has to be the moles per liter concentration of hydrogen ion in order for you to get the correct pH value. Other times you may need to actually work a little bit backwards. You may already have the pH, but you need to figure out what the hydrogen ion concentration at that pH is. So in order to do this is a little bit tricky because you have to have a pretty good understanding of how logarithmic functions work, but this is how you would do it. So if we set this up as an algebraic equation, we would say negative log base 10 of x, which is the hydrogen ion concentration that we're looking for, all of that equals a pH of 12.3. So in order to isolate x on one side of the equation, you would raise 12.3 as the negative exponent for a base of 10. So x equals 10 raised to the negative 12.3 power. If we do this, we get our hydrogen ion concentration of about 5 times 10 to the negative 13th moles per liter. And if you compare it to some of the numbers you saw on the chart on the previous page, this should seem pretty consistent with a pH between 12 and 13. So calculations like this you're not really going to do very often in a laboratory, but it is good to kind of know the mathematical operations behind how pH is figured. So let's talk about why pH is actually important. So we've already talked several times throughout a few of our chapters about biotech applications, making proteins, making all kinds of products that are meant to make our lives better and to make certain things more efficient for us. So many of those biotech products, molecules like proteins, may not actually function correctly if they are not maintained in a solution that is at the correct pH. If the solution is too acidic or too basic, the protein may fall apart on you, it may lose its activity, and if that happens, your biotech product is basically no good. So we need to understand what the deal with pH is. So the best example of this is when you consider enzymes. In this case, we're looking at two particular enzymes. One called pepsin, which is a protease enzyme that is produced by cells in the stomach. This is an enzyme that is best maintained at very acidic pHs, which is consistent with it being produced by cells in the stomach and being secreted into stomach acids. Very acidic environment, so obviously it makes sense that it would work best at very acidic pHs. If you compare that to trypsin, which is another protease enzyme, a protease being an enzyme that cleaves proteins into smaller fragments, trypsin is found in the blood, which we already understand the blood is at a pH of about 7.4 or so. Trypsin has a pH optimum of somewhere around 8, so it's not that far off from the 7.4 that it's usually sticking around. So if you look at this chart on the right side of the slide here, you can see the pH optimum in terms of the activity functions for both of these enzymes. So basically, the y-axis here is measuring the rate at which these enzymes can catalyze their respective reactions, and you can see that both of them reach peak activities at very different pHs. Pepsin, as we said, is best at a pH of around 3, which is very acidic. Trypsin is closer to around 8. So even though these two enzymes do very, very, very similar things in terms of what kinds of reactions they're catalyzing, they're very different in terms of what kind of pH they prefer. So if you worked in a biotech lab that produced both of these enzymes, you would have to take extreme care to make sure that both proteins are being stored in the appropriate solution that is at the appropriate pH. So I alluded to this a couple of seconds ago, but when proteins are not maintained at the correct pH value, the protein may lose its catalytic activity. So those two enzymes you see that right there may not work properly anymore if they are not maintained at the right pH. Or in more extreme cases, bad pH ranges for a protein may cause the protein to become what we call denatured, and it may totally unfold itself and become permanently damaged at that point. Most proteins will become denatured when they are exposed to very high temperatures, but extreme pH values will definitely do the trick as well. Okay, so practically speaking, in a lab, let's talk about how you go about measuring the pH of a solution. So we can do this one of two different ways. One of them is quick and easy, but it's fairly imprecise. The other is a little bit inconvenient, but you cannot beat its accuracy. So pH litmus paper is the first one that I mentioned, and a pH meter is the second one. So this litmus paper that we're talking about is going to be coated with a chemical that will change color in the presence of either an acid or a base. So based on the color change, you would dip one of these little sticks of litmus paper into your solution, and then you would compare the little squares to the kind of guidance chart that you see there, and whatever kind of best fits the color on your litmus paper that you use, that would indicate what pH you are dealing with. So this is, like I said, fairly imprecise, but it is good, it's quick and easy. It will give you a general idea of the pH of the solution, but I would not take it with anything other than a grain of salt. Now, the pH meter is going to be much more sensitive and much more accurate, but as I mentioned, it's a little bit inconvenient because pH meters are very sensitive, and therefore they require regular calibration with what we call standard buffer solutions. So in this picture, you can actually see a pH meter, and you can also see three different colored solutions here. The yellow one is a standard solution that is at a pH of 7.00. The pink one is at a pH of 4.00, and the blue one is at a pH of 10.00. So depending on kind of what range you are going to be reading in, you will need to standardize or calibrate the pH meter with two of those solutions, whichever ones are on either side of the pH value that you expect to see. So the pH meter works with a glass electrode, so you can see this little rod here that will have the glass electrode on the end, and then it is attached to the reading device via a wire that is connected in. So when making solutions, you are probably going to want the pH value exactly right. So this is going to be the instrument to use, inconvenient though it may be. Litmus paper is just not really going to do the job if you want your pH value to be a particular number. So there is a little bit of a extra methodology when you're making a solution that you desire to be at a particular pH. So we've already covered how you go about making a typical type of solution in simplistic terms, but if you need the pH of that solution to be at a particular value, there are a couple of extra steps that we need to consider here. So the first thing is that you need to adjust the pH before you bring the solution up to the final volume. If you get to the final volume before you ever consider the pH, you are going to end up with a higher volume than you should have, and that's going to mess up your concentrations. So you'll see how that works as we go along here. So as ever, whatever your solutes are going to be, you'll want to weigh those out. Then you are going to dissolve those solutes not in the full volume of solvent, but somewhere between 2/3 and 3/4 of whatever your desired volume of solvent is. So if you're making 1 liter of solution, you might want to just start with 750 milliliters of water. So that's going to leave you about 250 milliliters left to go. But as you're going to appreciate by the time we're done with this slide, that volume is going to come up a little bit as we adjust the pH. So you'll want to mix the solutes well in here in a beaker with a stir bar. The beaker, not because it is particularly good at reading volumes, just because it is good for mixing solutes in their solvent. And the stir bar is just a little magnet that you can put on a magnetic stir plate, and it will keep the solution stirring as you go throughout the pH adjustment process. So at this point, we are ready to start reading the pH. So at this point, we need to calibrate the pH meter. If we are expecting an acidic solution, we will want to calibrate with the pH 4 and 7 standards. If we expect something greater than 7, we'll want to calibrate with the 7 and 10 solutions. So we're going to suspend the electrode in the solution as it stirs. So even as the solution stirs and as we adjust the pH, everything is going to be mixing well together, and we'll get a good and accurate reading for what the pH of the whole solution is. So let's say that the pH of the solution is higher than what you desire. So let's say we want a pH of 7 and the pH is at 9. So if this is the case, we are going to want to add some acid to bring the pH down. So we could add dropwise an acid like hydrochloric acid, usually in its concentrated form. So we would add hydrochloric acid. That's going to increase the hydrogen ion concentration in solution, and we'll do that carefully, drop by drop, until we get to our desired pH. On the other hand, if the opposite is true and our pH is lower than what we want, it's too acidic, we would instead add a strong base like sodium hydroxide. This will add hydroxide ions to solution and reduce the hydrogen ion concentration, and this will get the pH up to where we want it. So those are the most typical acids and bases that you will use, but you can use other acids or other bases if you decide that, okay, I don't want chloride ions in solution, or I don't want sodium ions in solution. You can use other acids or bases as well. So once you have adjusted the pH, keep in mind you're adding acid, you're adding base, you're adding a volume of something to this. So this is exactly why we did not bring the volume up to 100% right away, because if you start adding acids and bases, you're going to go above that 100%. So if we started with, say, 800 milliliters, the 750 of water, and then whatever the volume of the solute was, if we are now at, say, 825 after adding our acid or base, now we can bring the volume up to our desired mark. So we'll add water, and then we'll just check one more time to make sure that the pH is still good, which it should still be. Okay, so that is going to do it for this particular video. So join us next time, and we will start talking about buffers in biotechnology applications. So buffers have everything to do with keeping the pH where we want it. So I hope you will join us next time. See you later. Bye-bye.